Apologize in advance. Although my work has been about brains and their evolution, human language, human consciousness, I will not say much about consciousness. You can ask me about consciousness later on. It's a little bit far afield. I wanted to stay today focused mostly on the first part of the question, and that is the question about the origins of life. Now, what is a neuroscientist, an evolutionary biologist, doing talking about the origins of life? The answer is studying the way brains evolve has been a real challenge. Brains don't evolve the way a lot of things evolve in the world. Brains are interacting with the world in a very deep way, and that makes it very much more a complex problem. And as a result, it's led me over the years to ask some much more basic questions. And one of them has to do with the nature of the origins of life. Why the origins of life? Well, it came to me initially in reading the closing lines of The Origin of Species. Darwin, in it, he cryptically acknowledges that the process of natural selection is insufficient to account for the core attributes of life. Now, when we think about Darwin's Origin of Species, we sort of think about it, this is an explanation of evolution. This is the whole story. Obviously, those of us in molecular biology and cellular biology and evolutionary biology know that, of course, it's only part of the story. But what's interesting is that we very seldom recognize that I think Darwin recognized this as well. And in fact, it's one of the few things that he vowed never to write about. And in fact, it's only in one letter that he wrote to a fellow named Asa Gray that he says anything about the origins of life. And I think there's a very good reason, and I think it's captured in the beginning of this last line from The Origin. There is grandeur in this view of life, with its several powers, having been originally breathed into a few forms or into one. Most people use the rest of this quote to start their books or their talks because it's all about, how evolution went on as the orbs went around and time went on. I'm much more interested in this interesting statement about the special powers. What are the special powers? Well, the story here is that natural selection doesn't explain the origin of these features of organism design. Natural selection is about how it gets honed. But natural selection doesn't work until there's something that has these powers. And that's basically what Darwin is saying. Now, interestingly enough, it's only in later editions that he has the Creator breathing them. Early on in his early versions, he doesn't have the Creator breathing them. I'm not going to talk about creators here. But actually, the real question is, what are these special powers? The important thing I want to say is they're not just the initial conditions for evolution to get started. We're talking about something that must always be there, something that must be very general. Now, the excitement about the origin of life, and why I think it's an interesting scientific and philosophical question as well, is because the origin of life had to have been simple. Some really simple process. It could not have been complicated. It had to be initiated by physical principles that we understand are not living principles. And yet, it created a fundamental change in nature. So what's so interesting about it is something simple created a radical change in the way nature runs, the way causality happens in the world. And that's what makes it, I think, a really fascinating question. And it also means that the origins of life question is not an evolution question. I like to think of it as an emergence question. It's a question about how evolution came into the world. It did not evolve. So I think that the question, what is life, is too narrow. Although it's a wonderful question, it focuses us on us and creatures that we understand, and the earth, and how the earth developed, and so on. I'm more interested in what you might call a universal biology today. I want to talk about life in general, in the big sense, big G general, how it might be everywhere in the universe, perhaps, and what might be these special powers. Thomas Huxley has an interesting way of making the same distinction. He says, living organisms arose on the primitive earth in a series of stages from non-living matter. This process can be called abiogenesis, to distinguish it from the concept of spontaneous generation. What's interesting here is that life had to ultimately have originated spontaneously. It had to have originated not by an evolutionary process, but in some other kind of process. And that's what I want to get at. Now, I want to say a few things. They shouldn't be taken as criticisms, but just to talk about some alternative ways we have of thinking about the problem. Obviously, we can take the approach of looking at early fossils. And this, of course, is not one that I have been following, but many people, including folks here, I've just learned some great stuff about it, in which you can study very early life, getting close to the origin. The problem is, if the origin of life is very, very simple, that probably won't fossilize. We probably won't see much of a signature. How do we go back farther than that? Well, there have been a number of sort of experimental ways. And to sort of tongue-in-cheek, I like to call one of the most common ways we're doing it today, in terms of molecular science, I call it the Frankencells approach. Now, as I say, it's tongue-in-cheek. This is excellent work. I don't want to make fun of it. But what's going on in this is we're recombining components that we extract from once living creatures, from cells, typically simple cells. Maybe it's DNA, maybe it's lipid membranes that form the surface of cells. And then we take these things from once living cells, now separated in our test tubes or whatever, and we put them together in interesting ways and ask the question, can we reanimate this complex we've put together? Well, that's the Frankenstein story in cellular molecular biology. In effect, can we take bits of something that was living, and just bits of them, not the whole thing, put them together in just the right way, and will they come back to life? We can learn a lot from this, and I show two very, very prominent examples here, in which we contain a little DNA activity inside of a bubble of lipids, fats, a little bit like cells work. Cells have a lot more in them than just DNA and a few other molecules. These are DNA molecules stuck on the surface of clay, interestingly enough. Other ways are creating sort of lipid bubbles with molecules stuck to the outside of these. All of the parts are parts that you would take from living cells and put them together in interesting ways. So it's the Frankencells approach. There is a sort of logical requirement approach, and it's taken from many different directions, from physics, from computer science, and from evolutionary biology. And I list just a few and how they think about it. Erwin Schrödinger, one of the major figures and the father of quantum mechanics, wrote a book called What is Life? And in it, he picked out two things that he thought a physicist should know and worry about when thinking about life. One is what he called the problem of negentropy. How is it that organisms seem to beat the second law of thermodynamics? That's a really odd thing, because most systems left to themselves will, as you know, become messy. They'll fall apart. They won't maintain their integrity. The second one is information. Life reproduces itself. It passes on some kind of information, some kind of instructions from generation to generation. In the 1940s, when he was writing this, both were sort of mysteries. His thinking about the information component, in which he said it's sort of like an aperiodic crystal, a crystal that's got repeated parts, but they're not exactly the same. You need something like that to do information. That view was absolutely critical for Francis Crick in his thinking about the DNA molecule and how it might work. So a physicist made a very significant contribution. What's interesting is that these two sides of it, the thermodynamic or entropy side of the problem and the information side of the problem, for the most part in history since the 1940s, have been following along in separate domains. And people doing the work in these areas are oftentimes in very different departments doing different things. A friend of mine, Stuart Kaufman, also very interested in this, is interested in how information gets copied, replicated, but also that in fact cells do what he calls work cycles. A work cycle is something like your automobile engine does. It kicks itself off, but it has enough power to get back to where it started again so it can do it again the same way. Life has to have a kind of a work cycle logic to it. It's part of the entropy problem. It has to do work to repair itself constantly, but it has to come back to where it started so it can continually do it again and again and again. An interesting and little understood theory was put together by a man named Tibor Ganti in Hungary back in the 1960s. He called it the chemoton. Basically, he said, look, let's add one more thing to this. It's information and the work of metabolism, but it's also a container. You need a container. Things need to stay in one place. Richard Dawkins, the well-known evolutionary biologist and sometimes writer of popular and maybe even exaggerated books about evolution, thinks about evolution, that the origins of life might have been what he calls starting with a naked replicator, some molecule that just got itself copied somehow, and that's all you need to start life. I'm going to suggest that that's, in fact, not very helpful as a starting point, but it's an interesting one. It picks out one of these aspects. And the point I want to make is that people have been picking out these different aspects. It's a different way of doing sort of the Frankencells problem. It's saying, it's a kind of a reverse engineering logic. I know what life is like now. I know what it uses now. Let me see these parts and see if I can put it back together and see which ones are most important, which comes first, which comes second, and so on. One of my very favorites is not a biologist at all, not anybody interested in the origins of life, but was one of the fathers of modern computing machinery, John von Neumann. Late in his career, just before his untimely death, he spent a lot of time working on what he called self-reproducing automata. That is, machines that reproduce themselves. A really interesting idea. He felt that, look, if life does it and life is a kind of machine, why couldn't we come up with an engineering trick that did the same thing? Interestingly enough, we have not done that yet, even though he went far into it. And here's a sort of depiction of his process. You've got to have a description of yourself. You've got to know what you're supposed to look like, what all your parts are. You've got to have this description. And then you've got to have another set of description, which is what to do, how to assemble it, how to get there first. So this is the information side of it. You've got to have your self-description and your assembly instructions. And then, a computer theorist, he said you've got to have a machine that builds it, that takes these assembly instructions and recreates the self-description. He did leave out a couple of things. You need some energy to do this, and you need a lot of raw materials. He wasn't worried about this. He said, well, we can maybe just add this in. He found that it was easy to describe the informational problem, but really hard to describe the physical problem. How do you actually do it? It led later on, and in recent times, to an area that's sometimes called artificial life or A-life, where we find lots of computer algorithms that are doing the information side of John von Neumann's story in cellular automata and things like that, but don't do the physical process. For our purposes, the physical process, I think, is really crucial. So here's just a couple of reverse engineering quotes, but they have an interesting challenge in each of them. The first one by J.B.S. Haldane, one of the fathers of modern evolutionary thinking. He says, a simple organism must consist of parts A, B, C, D, and so on, each of which can multiply only in the presence of all or almost all of the others. In other words, there's a kind of a synergy that's necessary to life. James Strick, fairly recently, criticized a lot of theories about origins of life and how we think about it. He said that if metabolism is dominated mostly by proteins, but is a prerequisite for the functioning of nucleic acid information molecules, how can a system like our current living cell, even the simplest prokaryote, like a bacterium, with each of these two parts totally dependent upon the other, ever have evolved in the first place? So this sort of is the counter to the point I made at the very beginning. Life must have started in an incredibly simple way. And yet it looks like when we try to reverse engineer life, these components, how could we get such complicated things together? That's really the challenge I see that's out there. And I see this as a challenge not just as a challenge for biologists or even evolutionists, but a challenge as a sort of a basic challenge about how something so simple can produce such a difference in the physics of the world. This is a complicated picture because I'm not going to explain a lot of what it says. But let me just clue you into, I'm going to walk you through in a few steps what I think are one way to approach this problem. This has to do with thinking about dynamical processes, not the stuff of life, but exactly what life does, what an organism is doing, the changes that are taking place. Because my focus will be not on the stuff of life, because I think the stuff, the molecules, are not the critical piece. I think what's happening is a change in the way causality, in the broadest sense, actually takes place. That in living things, things work differently. Information and the ability to resist the ravages of entropy increase are critical changes in the physics of the world that occur with the origins of life. And so I think these need to be understood in what I would call dynamical terms. I've coined three terms. Actually, it turns out that each of them, except for the latter, the last one, have been coined before for related things, interestingly enough. We never can be fully unique in our thinking about these things. Somebody's always sort of preceded in some piece or another. Homeodynamics refers to dynamical processes that even things out. That's homeo. So when we think about equilibrium, things are basically equaled out. It's sort of the egalitarian state of things when they've relaxed in the world. Thermodynamics is the classic way of talking about it. I wanted a general term that talked about it outside of the realm of thermodynamics. We could apply it at the level of perhaps quantum events, or we could apply it at the level of social processes. I wanted something to talk about a dynamics of evening out. Thermodynamics is our model system. What happens is constraints or asymmetries get eliminated in this process. They happen spontaneously, and things become more and more evened out. Morphodynamics, a second piece of it, and you'll notice that it's nested within in this picture here. That means that it requires thermodynamic or homeodynamic background, but it's a special case. Morphodynamics is sometimes called self-organization, and that's what I'm going to talk about a lot today, is this process we call self-organization. Under these circumstances, and I'll give you some examples, what happens is processes that look like they should be getting more messy. We, in fact, are perturbing things all the time in these cases, upsetting things, throwing new materials into something, shaking them up, and so on. And what happens is we find that oftentimes continual perturbation actually organizes things. You might think it should make things more disorganized. Oftentimes it can organize things. And that's one of the exciting new areas that in the last couple of decades has been explored by lots of different fields. It's also the bridge to what I really want to focus on, which is the third of these, which is a special case of the morphodynamic processes. Morphodynamics define morpho referring to form. That is, things become more formed, more orderly over time instead of less so. And finally, with life, evolution, semiosis referring to thought processes, communication processes, information processes in general. What we find are things are organized with respect to ends. They go in particular directions that are prefigured, so to speak. In none of the other processes that I've just talked about is there a kind of prefiguring going on, a kind of memory or information or kind of representation process. But living processes and mental processes all involve this kind of thing. And so the question is, this is that transition I've been talking about, the transition from things that look like they should get more messy, in which information in the sense of representation makes no sense at all. In physics, we don't need those terms. But in life, we do. We have to talk about adaptations, about functions. A function is something that has an end. It accomplishes something. And it accomplishes something for something. It achieves something with respect to a subject. I want to use some terms from complex systems. I'm going to use them in a much more generic way, just to sort of get a couple of ideas across. The complex systems theory has talked about attractors oftentimes. I won't go into the details of what an attractor is. It is not something like a magnet that pulls something. It's a process that becomes statistically very, very predictable. And I'll give you some examples of that. An attractor is something that if you've disturbed a process, but then let it go, it will tend to go back to a kind of a ground state. That ground state is oftentimes chaotic, but it's not all of the possible ways things could vary. And attractors, in that sense, are, you might say, stochastic in the scientific terms. That is, they're a little bit chaotic and a little bit organized at the same time. The simplest version of an attractor, we don't tend to think of it because we've got different names for it when we talk about thermodynamics, is this one, why things fall into a mess spontaneously. And what happens is we find in the natural world that, at least in the non-living world, what we have are things that are unevenly distributed and organized, as we might say, and over time they spontaneously become more evenly distributed. That happens all the time. This is what the second law of thermodynamics is about, why the world tends to fall into mess spontaneously. And we know this from our own lives, of course, as well. What I want to point out, however, is that you can put these back together like this, and we all know how to do that. It takes work. In order to keep your room clean, in order to keep all the papers piling up on your desk organized, you have to do work. You have to do something to make this occur. And so, to some extent, what the concept of work has to be understood as the inverse of this. You have to do something. From the outside, you have to impose on the system in order to move in this opposite direction. And so I have this kind of little funnel picture here to give a sense of that asymmetry. When things are relaxed, there's no work being done, they tend to go this way. Constraints, that is, things that keep things in regular, uneven distributions, are lost in this process. I've given it a term so I can talk about it in very generic senses. I call this orthograde. From the old hippie days, this is going with the flow. If you're orthograde, that means you're in the same direction as the tendency of things. If, however, you want to push things back in the other direction, you've got to go against that flow. You've got to swim against the grade, so to speak. And so I've called that contragrade. Work is always producing something that goes against the flow of something. There's a number of natural tendencies in the world that are attracted, so to speak, towards these, not regularities, but in a sense, asymmetric targets. No matter how you start out with an organization here, shake it up a little bit and you're going to end up here. Now what I want to say is the same kind of logic can be seen at higher levels. And I want you to sort of see this in abstract before I go any further. First of all, in morphodynamics, the simplest example are things like whirlpools. A whirlpool is something that is there because it's constantly being perturbed. Either something is draining the water out, or there's a stream of coming downhill and there's a barrier, and the water moving around the barrier becomes disturbed in its directions of movement. And initially you drop your rock into the stream, and initially it's very chaotic. Initially following where the rock was dropped in, you're going to see a lot of random movement of the water, what we call the rapids and so on. But over time, as if the flow of water is constant, it will very often regularize. And it regularizes into this kind of circular symmetry that we're all very, very familiar with. Interestingly enough, of course, at each second, it's different water. It's not the same water. The only thing that stays constant is the flow, you might say, the symmetry of that flow. What I want to point out is with a system like this, you also have something like an orthograde and contragrade effect. First of all, to produce it, there has to be work going on. That is, in other words, there has to be water continually pushing water out of the way, and a rock continually perturbing the motion of that water. That's doing physical work in that process. In fact, what happens over time is that the chaotic flow that's following this, those water movements that are in effect not in correlation with each other, but as you might think about it, bump into each other like a crowd trying to go across you've probably seen this on crosswalks. When there's a lot of people on both sides and they come together, the initial collision is not a very pretty one. People are sort of jostling around, but pretty soon they typically form into flows that they pass each other. Those things that are not in alignment tend to slow each other, sort of block each other, interrupt each other, cancel each other. And it's those processes of movement that are consistent with what amounts to the least, what I call sometimes the least discordant pattern of movement are what's left. Now, what's interesting about a whirlpool like this is that it has an attractor state, an orthograde tendency, but of a higher order sort. If I go in and stir this up, but then pull my oar out, over a very short period of time, it will be chaotic and then come back to a regular flow again. In other words, it has a tendency to go back to not an equilibrium kind of state, but a dynamical state that's quite regular. And what's happened here is it has a new kind of orthograde tendency, a new kind of spontaneous tendency to, when it's disturbed in any possible way, to go back to this sort of ground state. Now, finally, what I want to say is living things have something similar to this, but again, it's of a higher order. And it's important to see this transition. Living things also disturb them, and they tend to go back to something. What do they go back to? Well, you and I are not stable either. We're continually taking things in and getting rid of stuff. And our bodies are being constantly replaced and replenished, a little bit like a whirlpool. But we're changing also. We're not staying the same. We have a lifespan. We develop, we mature, we age, and finally the whole system burns out. And yet, when we get hungry, when we don't have enough food coming in, we pursue it. When we're in dangerous places, when we're in too hot, too cold, too damp, too sunny, whatever, we adjust. Living processes adjust to this. Whirlpools don't adjust. Living processes do what we call adaptation. They adapt to their context in a way that maintains their dynamical attractor. But their attractor is sort of a moving target. It's the state that they come back to as a kind of a lifespan state. Now, there's one thing about inorganic versions of this process, like the whirlpool. They have a property that's called maximum entropy production. Now, it sounds very, very technical. It's not all that difficult to follow. One of my favorite examples, in fact, is one of the favorite examples of people in the complex dynamical systems work looking at inorganic processes, is this sort of thing. This is actually a sort of depiction of something that shows up in a thin surface of oil on top of a heated plate. What happens is if you heat oil up fast enough, it's of course giving off heat into the air around it. But as you heat it up to the point where it can't get rid of the heat as fast as you're putting it in by molecules randomly bumping into each other, we call that conduction of heat through the liquid, something begins to happen. As the heat begins to accumulate asymmetrically, it causes the hotter areas to become lighter because they expand. And they rise up to the surface faster. And those areas that are on the surface that are a little bit cooler and have given up some of their heat begin to fall. And what happens is regular motion begins to develop. And here I show it as hexagonal cells. It literally from the top looks like a bunch of beehives and honeycombs. Regular cells develop, regular hexagons develop of moving fluid with hot stuff going up through the center and the cooler stuff going down around the edges. Hexagons are formed in a solution that has no other reason to be hexagonal except that this is the most efficient way to get rid of the heat as fast as it comes in. We call this maximum entropy production. It's giving off its entropy, that is, it's giving off its disturbance into the environment as fast as it can in order to keep up with how it's being constantly disturbed. You can see it's a little bit like the whirlpool, being constantly disturbed and having to find the most efficient, the most work-efficient way of giving off that disturbance. We find this in lots of other examples. One of my favorites, and around here where I live, I don't get to see this much anymore, but our snow crystal formation. Snow crystals have this wonderful hexagonal shape. It's in part because water molecules have just a few ways of forming into sheets or lattices. But also because as the snow crystal falls down from its initial development up high in the atmosphere, one of the things that's happening is it's randomly accruing water molecules to its surface. And the shapes that they grow tend to be shapes that are determined by the pressure, the temperature, and temperature and the humidity. As a result, they grow outwards and they freeze the pattern that was just before that. But each time they freeze that, it limits what next can happen, what's next most likely. And that one then limits what's next most likely, which limits what's next most likely. And so they become, in effect, a historical record of this process in which you're continually perturbing it and it's regularizing itself by what I would call compounding constraints. Constraints are reductions in the degrees of freedom, reduction in the possibilities of changes or additions or modifications. As the snow crystal grows, it's limiting what can happen next. And its shape plays a significant role as it grows. It looks like I'm going to have a problem here. This seems to go in and out. Somebody with another one of these may have to come up at some point. Here, I'll take that just in case. Got it. Okay. The difference is that snow crystals stop this process. They capture it. They take sort of a snapshot of its history, and it's captured there. Unfortunately, they don't get to reproduce. They've acquired all of this regularity. They've built up their constraints, And it's the building up of constraints that produces order. Constraints are reducing all the possible variations. Well, if you think about it, what we mean by order has to do with the same thing, reducing variation. The more regular something is, the more repetitive it is, the more symmetric it is, the more we think of it as orderly. And this is how self-organizational processes do it. They do it to minimize the total path by which disturbances get eliminated. But in so doing, they regularize to do it. Now, that's going to become a very important part of the story. It turns out that a lot of these processes are what we call nonlinear processes. It's the best example of nonlinearity is your credit card. Your credit card, if you don't pay off the balance every month, you'll get charged not only for what you had last month, but also this interest charge goes on there. And next month, you're now paying off also the balance and the interest. You let that run too long, and the interest begins to outweigh the balance by quite a bit. This ballooning mortgages and things are like this problem. This is nonlinearity in a simple sense. And what it is you're compounding things. You're building, you're adding things up so that each time you do the next cycle, what happened in the last cycle has influenced it a little bit. But as you can see, that can build and build and build and build. Well, that's effectively what's going on in what we call self-organized, or I like to call morphodynamic processes. Now, most morphodynamic processes in the non-biological world are very fleeting, or in fact take a lot of work to produce, like laser light. I won't go into how it's produced and why it's like this. But in effect, it takes very special circumstances, in part because the process of getting rid of disturbance as fast as possible is involved in destroying its own basis. And that's what I want to get to next. So let's think about this nonlinear compounding of constraints. Here's a couple of snow crystals. You can see their wonderful form. It's interesting that very recently people have begun to do scanning electron microscopy of snow crystals. Way to do it. Usually you can't do it because you've got to have things hot, but they've figured out ways to keep things frozen. It turns out that microscopically, at the very ultrastructure level, they're very noisy. They're not regularly organized. But at the billions and billions of water molecules that form a regular lattice, they can become regularized. They become regularized in large numbers because of a kind of a statistical process that's involved here. And what you can see is that across as this grows out, you see that each of the tips is growing similarly. The symmetry is there in part because heat is distributed evenly as it cools off, and the constraints are growing evenly as it grows. What about life? Life also captures constraints. It builds up regularities and somehow captures them, but obviously it can't freeze them. And so the problem is that it's got to be a little bit like this, but a little bit different too. Just to give you a sense that these three levels, I call them emergent levels. They each emerge out of the other. Homeodynamics, that is, thermodynamic processes, were the basis of some of these self-organizing or morphodynamic processes. And I'm going to argue in a few minutes that relationships between morphodynamic processes are necessary to generate life. Life is not morphodynamic. It's not just self-organizing. In fact, the term self-organizing, I think, is very misleading. There is no self in any of these things I've talked about that's doing anything. But one of the things that's happening is, and this is for the physicists in the audience maybe, is that the way constraints and entropy are being moved through these systems undergo reversals at each of these transitions. And it's those reversals that are interesting. Going from a simple thermodynamic system to one that's dissipating and organizing, we see not that patterns are breaking down, but that regularities are appearing. Reversing this tendency, it's as though you played the movie backwards in that sense. And when we see living processes, we see, yes, they're developing regularity, but they're not throwing out entropy. They're not unloading this disequilibrium as fast as a system that self-organizes. In fact, they can't. They have to maintain themselves. So at each of these levels, there's a kind of reversal of a variety of dynamical tendencies. And I was trying to get at what that final reversal is about. That is what I think is behind what I would call the several powers that all life-like things have to utilize. A person that way back, a few hundred years back, had some interesting ideas about this is the philosopher Immanuel Kant. In one of the last things he wrote, he called it was the critique of teleological judgment. Wrote towards the end of his life. As his other critiques, those of you who know about Kant, he sort of plays with, he's like his own debater. He's playing with the opposite sides of the argument and debating against himself and coming up with a sort of, you might say, the mean argument in the middle. Well, he does this also with the problem of teleology, with purposefulness. Teleology is one of these problems for those of us in the sciences because we have no need for it in the physics and chemistry. But suddenly, it's something like teleology seems to be going on in biology, and certainly in mental processes. And it's troublesome how it is that a physical process that doesn't have any hint of that is giving rise to something that does. Should be the kind of thing that we might want to, bring in some divine designer to make sense of. And, of course, many people have. So Kant distinguishes between organisms and machines in interesting ways. He says a machine has solely motive power, whereas an organized being, means an organism like you and I, possesses inherent formative power, and such, moreover, as it can impart to material devoid of it, material which it organizes. This, therefore, is a self-propagating formative power. I read formative power as producing constraints, as generating regularities, forms, organization. He then goes on to say it's a self-propagating formative power in which every part is reciprocally both ends and means. Now, you can see in this last clause something I mentioned from Haldane early on with A, B, C, and D all helping to reproduce each other as to define an organism. Kant uses the phrase and comes up with this notion of self-organization in talking about organisms. And when he says that every part is reciprocally both ends and means, he means that you need some molecules to make these molecules make those molecules, and these ones make the first ones, so all the parts are made by parts inside. Calling it ends and means is a problem, and in fact he debates himself on this in this argument. I won't go into Kant's philosophy about this, but he basically says when we look at organisms, it looks as though they have intrinsic teleology, intrinsic end-directedness. He then backs away from it and he says, yes, but applying the term ends and means basically is a perspective I've taken on it, and it's not intrinsic. What I'm going to suggest is that, in fact, in that sense, he's wrong. In that sense, that it really is intrinsic. But it's not quite the same as he suggests here. It's a little more complicated than this. So when he says that basically the formation of these components, the building all of the parts, a system that builds all of its parts reciprocally in this sense resembles teleology. But in fact, that's not what causes teleology. No matter how you look at it, that would still be a kind of outside perspective that sees the production of these things as the ends and other parts of it as the means. Along this line, a number of researchers, beginning with Francesco Varela and Umberto Maturana, came up with a notion, again, another way of sort of stating what Kant had to say. And they call it autopoiesis. I won't try to break it down. My way of decoding that is to say it's self-fabrication. That's what Kant is talking about, basically, a kind of self-fabrication. So here's Francesco Varela's way of saying an autopoietic system is a minimal living organization. Notice it's very close to what I'm trying to talk about here. Is one that continuously produces the components that specify it, while at the same time realizing it, that is the system, as a concrete unity in space and time, which makes the network of production of components possible. If you got lost in the middle of that, that's okay. It's pretty gummy. The problem is that autopoiesis describes what must be true about organisms, but it doesn't explain it. It's not a scientific story. It's not telling us how it works, so to speak. It's saying this must be true. Organisms have to do this. And an additional point is that simply having components that make other components that make other components doesn't define a unity. Varela says it has to do this to be a living organism. It has to be something that unifies the whole. But it doesn't really tell us how that happens. What I'm going to try to do is to answer each of those with a fairly simple sort of thought experiment that is also a potential scientific experiment. Here's a process that has this reciprocal ends and means. Sometimes it's called reciprocal catalysis or autocatalytic sets. Autocatalysis means catalyzing yourself. A catalyst simply is a chemical reaction that makes other chemical reactions much more probable and can speed them up sometimes thousands of times. In this particular case, there's a molecule E which hits a molecule A, and because of its shape, causes it to break just right to become a molecule C and B, and C hits another molecule D, and just right, because of its shape also, breaks it up in just the right way to make another molecule E, which then can start the process over again. This is a reciprocal catalytic process. The catalysts are here, and they're causing a reaction here. The yellowness, if you want to look at this, is basically energy. When you typically break bonds in a molecule, you release some energy. And typically the parts carry that energy away. So this thing can also self-fuel itself. This kind of a process can fuel itself and continually run and run and run. And in fact, I've only shown one cycle around this, but notice that I've gone one cycle around. This thing didn't get lost in the process. What happens is you begin to multiply the catalysts, and the process goes faster and faster and faster. And like other self-organizing processes, can very rapidly produce big asymmetries, for example, in concentrations. And there's some interesting, though I won't talk about them, some interesting chemistry behind this, and people have done nice experiments that kind of demonstrate this process. But this is a process in which there's this kind of reciprocal production, reciprocal formation. Notice that there's no individual here, there's no unity here, and in fact, once this thing goes, the stuff will diffuse away and we'll get more and more catalysts and more raw materials. This is co-production, it's reciprocal co-production, reciprocal formative power, you might say. But there's no unity that holds it together. Nevertheless, a number of people have hit upon this because it looks a little bit like reproduction. You're making more of yourself, or more copies of things, so there's more E's showing up in the world as this process runs. It looks a little bit like reproduction. So it's one of the areas where people have sort of focused on maybe something like an autocatalytic set or a reciprocal catalytic process might be a sort of early starting point for living processes. Little cheap animation there. I just want to distinguish between autocatalysis and reciprocal catalysis. Autocatalysis is in which a molecule A increases the probability of more A's being produced. This does happen sometimes. It's fairly rare to get a molecule that can do this. Reciprocal catalysis involves a collective, more distributed version of it. Autocatalysis might seem a little bit more like self-reproduction. You don't need anybody else to help you out. You're a little molecule that makes another version of this. We've run into a problem like this in recent medicine and biology with what are called prions, molecules. Those are associated with what you may be familiar with as mad cow disease or Creutzfeldt-Jakob disease or Kuru, depending on where you come from. These are proteins that once they get into your nervous system, they find other proteins that have the same chemical makeup but a different shape. And they come in contact with them and change their shape. And so what we have is the nervous system is constantly producing what we call pre-prion proteins. That are used for very important features of the nervous system function. It's beginning to become clear that they may play a role in memory consolidation, for example. And that's why brains produce them. But it turns out they periodically get bent into an inappropriate shape that we call a prion. And a prion takes the pre-prion proteins and bends them into that shape as well. That's a kind of autocatalysis. It's not making something new, it's just changing its shape. But it's making more prions out of preprions. The problem is that preprions can be broken down and remetabolized into the nervous system. Prions can't be. And so you produce a lot more of them. They just become junk that fills up the nervous system and causes trouble, as you might expect. We also have fairly well understood examples of what I would call reciprocal catalysis. PCR machines that amplify DNA are basically doing reciprocal catalysis. There's a string of DNA that is stuck together with another string. We heat it up, they come apart, and each one of them can now produce the complementary other string. So we get an A that produces a B that lines up on it, and a B that produces an A. You heat it up again, you spread them apart, and you produce more A's and B's in this process. This is a reciprocal process. It's not autocatalysis. So some people like to think about DNA, Richard Dawkins in particular, as a molecule that replicates itself. No, it's a reciprocal catalytic process. But of course it involves much, much more than that if you don't have something to heat things up and cool things down in the right way. There's a couple of things about both of these processes. In any of these kind of reciprocal catalytic processes, there is no self-maintenance. There's nothing that keeps the system together. There's nothing that says that I've got all these catalysts, they're always working together. These are just things floating around in solution. There is nothing that's also far from equilibrium. The catalytic process is something that rapidly pushes things towards an equilibrium. It sends things in a particular direction. Living things are kept far from equilibrium. We're sort of, like Alice and the Red Queen, running as fast as we can to stay in one place. That's far from equilibrium. That's not happening here either. There's no self. There's nothing that tries to maintain itself, to hold itself together here. They lack the qualities that organisms have. But more importantly, all self-organizing processes destroy their very basis, and they do so as fast as they possibly can. And that's a real problem. These are optimally offloading any kind of perturbation, offloading precisely the conditions, the gradients of, say, heat or molecular types that set them in motion. And they do so faster and faster and faster because they have this kind of snowball effect. Life can't do that. That's one of the great challenges. Life cannot do that, and it leads us to a paradox. Because nevertheless, life has to generate regularity. It has to generate structures. It has to generate its order because it's continually having to deal with the fact that it's breaking down by virtue of thermodynamic decay. And so we've got this interesting paradox. Living systems must depend upon morphodynamic processes to generate the constraints, that is the order, required to channel the work that prevents thermodynamic degradation. And yet, living things must ensure that these processes don't undermine the very conditions that they depend on for their persistence. In other words, the self-organizing problem, you might say the darling of complex systems theory, has not quite yet crossed the threshold to solve the problem that life had to solve, and that is to persist itself and to do what Schrödinger said, that is to maintain itself far from equilibrium. So another way to say it is that organisms rely on self-organization for constraint generation, but they invert its logic. They invert the logic of self-organization. That's why I say you've got another flip to take into account. And I'll try to explain how that works. So the main thing is that organisms have all kinds of autocatalytic-like processes in them. Circles of catalysts producing other catalysts, molecules producing other molecules, but they have to be more than that. And the question is, what's the more than that? How to talk about it. This is part of one of the several powers, but it's not enough. And so what organisms do is they're organized. Their very organization is set up to do just the reverse, to counter or compensate for this loss that self-organization itself produces. It's an interesting paradox. Self-organization is destroying its own basis, and yet living systems have to use self-organized processes, and yet they have to also not allow that basis to be destroyed. That's the interesting paradox that's here. And organisms, in fact, when we actually look at what's going on, there's been an argument that for many of years between various sides of this, some people still think that organisms do maximize entropy production and are hoping that it's going to be self-organization all the way down. I think the evidence is now beginning to undermine that story. But I won't go into the evidence. I just want to talk about the general principles now. Because what we see is that when we look at how organisms interact with gradients in the world, interact with energy sources, food sources, and dangers, and so on, is that they organize to increase the indirectness of how energy flows through them, how materials flow through them. By indirect flow through, they can use it to do lots of little kinds of work. It's like siphoning off the stream as it's running downhill to do different things. Getting a little bit of it, using it. Of course, it's going to end up down at the bottom of the hill. But you can, if you run off these little rivulets to do different things, you can do lots of different kinds of interesting work. Life excels at doing this, at finding interesting ways to get in between these sort of gradients and utilize them as best they can to take advantage of it. On the other hand, there's sometimes in living processes when, in fact, you need to grow really fast, and you do it oftentimes by really taking a lot of energy, a lot of materials, and being very wasteful about it. So life can do both in different contexts. There are contexts in which it may want to do this. Well, there's another process that I want to talk about to close this loop, because I think this is, in at least living processes, the other half of this problem. And that has to do with what's called self-assembly. Self-assembly is like crystal growth. Crystals grow because molecules come together because they have a particular symmetry with respect to each other. And as a result, they pack together in regular cells. And if they're all the same kind of molecule, whether it's salt or sugar or something more complex, what you get is regularity that shows up. But in living systems, there's another kind of crystalline-like growth process, and it's typically called self-assembly. And this has to do with molecules that have a shape that also allows them to fit together. Sometimes, as in cell membranes, because there's hydrophobic and hydrophilic components to it, or sometimes like in viral capsules, because you've got proteins that have very regular shapes that tend to stick together in just the right way to create enclosures like a viral capsule here. Inside cells, we also see it. This is one of my favorite animations. These are microtubules, a molecule called tubulin, a little two-part molecule. In certain conditions in the cell, becomes in effect sticky. Hydrogen bonds make them stick together, not as tight bonds, but just sort of, you might say, slightly weak bonds. And at other times you change the concentration, typically of the salts or the heat or whatever, and you can cause these tubes to shut down. Tubes are containers also, and I'll come back to talk about both these kinds of containers. Here we see a cell showing all the microtubules in it. What you can see is it's kind of like the skeleton of many cells, particularly eukaryotic cells. But tubes are formed in all kinds of interesting ways. And tubes and you might say polyhedrons, if we want to call this a polyhedron, are sort of the major ways. And sheets, like here, like in cell membranes, are the major ways that self-assembly takes place. Self-assembly answers the question that von Neumann was struggling with. How do I get a construction device that takes the instructions and builds something? Well, in organisms, you don't actually have to instruct things to get built into structure. Oftentimes, you can take advantage of self-assembling processes. And all you have to do is to adjust the parameters that make the self-assembly more or less likely. Well, what I'm going to do is to put these two together. And I want to show something interesting about these two features, autocatalysis at the top, or reciprocal catalysis, and self-assembly. Because it turns out that they each are producing constraints. They each are self-organizing or morphodynamic processes that, for a brief period of time, amplify constraints, make things more regular in the process of getting rid of entropy. But interestingly, their boundary conditions are exactly inverted in interesting ways. So, spontaneously self-amplifying catalytic chain reactions produce high local asymmetric concentrations of a small number of molecular species. In other words, autocatalysis, because it's self-amplifying, builds a local concentration of just one kind of molecule or a couple of kinds of molecules very, very fast. Eventually, they diffuse away. That's the problem. It requires a limited diffusion. You can't let the catalyst diffuse away too often, too fast, because they depend on each other to keep running. But of course, diffusion wins in the end in most cases. Self-assembly is interestingly different. It requires a lot of the same molecules close by. In order to grow like those microtubules I showed you, you've got to have a lot of tubulin molecules nearby. And they've got to be there in just the right time to sort of go together. If you have lots of diverse molecules, it's very hard for self-assembly to take place. So if you want to grow crystals in your home, saturate a solution with salt or something. And make sure there's not a whole lot of other kinds of molecules in there, because otherwise the crystals aren't going to grow normally. You've got to have a really homogeneous solution for this to happen. Well, that's one of the things that's produced by autocatalysis or by reciprocal catalytic processes, high concentrations locally. And in fact, the other thing that has to happen for this to occur is that as the system grows, as self-assembly grows, it steals molecules from the solution. And therefore, it'll slow down and slow down and slow down. Wouldn't it be nice if you were continually producing more and more and more as fast as you grew? Well, in effect, that's what reciprocal catalysis does. It produces more and more in the same place. Finally, when you create an enclosure, whether it's like a viral capsule or a tube or some other kind of enclosure, it does something else. It stops diffusion. And that's precisely what's necessary for autocatalytic processes, for reciprocal catalytic processes that have a number of parts, to persist and to continually operate. You need to have constraint. It has to stay in one place. So they have, in a sense, each one has different boundary conditions and different things that it produces that have an interesting relationship to each other. And so we've come up with a couple of thought experiments that we're now in the process of, at this stage, just trying to convince some of our chemist colleagues to pursue. One involves a kind of viral-like model and one a kind of a tube model, in which, in this case, one or more of the products of the catalysis becomes self-assembling. Is a molecule that has a structure that builds a sheet or a tube or an enclosure. But what that means is, as the catalysts here in red begin to interact, in the same place that there's catalytic activity going rapidly, there's also containers being built. And what that means is, when the containers eventually close up, they're likely to close up around catalysts. Now what that'll do is that will eventually stop the process. But now you have an enclosed process, an enclosed system, that if it gets broken open somehow, will release the catalysts that will produce more catalysts, produce more growth of enclosures, and close itself again. This is a self-mending or self-reconstituting system because of this reciprocity between these two processes. In other words, we've got two morphodynamic processes, self-organizing like processes, that have exactly complementary effects. And that allows a system that has the capability to replace itself, to fix itself if damaged. If the constraints that hold it together are dissipated, it will tend to reconstruct those same constraints again. Now, the other thing about it is that there's a funny kind of outside-inside relationship here. Self-assembly generates intrinsic constraints, that is, enclosure. Reciprocal catalysis generates reciprocal constraints, intrinsic constraints over time, more of this concentration difference. It turns out that this is the boundary condition, the necessary conditions to get self-assembly to work. And self-assembly produces the necessary conditions to keep catalysts together so they can do this process. In one sense, this becomes the environment for this process, and this becomes the environment for this process to take place. They're supportive environments, but notice it's a kind of a closure of supportive environments. Now what we have is a system that has a kind of codependence between these two morphodynamic processes. But precisely because of this codependence, we have a system that can now maintain itself. Even if broken apart, it can put itself back together. And if broken apart severely, distributed out, might be able to reproduce itself. That is, spread them out so that they enclose now separately, and you can produce 2 or 3 or 4. There's a kind of a work cycle to this process. I won't go through this kind of process. The container gets breached, self-assembly starts up, and reciprocal catalysis starts up. And one of the things that happens is that the system will close and stop catalysis and stop self-assembly. This little simple model system I've described doesn't allow either process to run out. This process doesn't allow you to use up all the potential that's in the environment. It stops itself before it destroys its basis. That, I think, is another crucial feature. It's a kind of a ratchet. You're probably all familiar with ratchets. Ratchets are these things in which, you can even randomly move this gear around, just jiggle it, bump it a lot, but it will mostly only rotate in one direction because of the way it's set up, because of the asymmetries of the constraints, that it constrains some movements and makes other movements more likely. We're all familiar with other kinds of ratchets, hooks and things, but barbs, things with barbs like bee stingers, have this same feature. Random activity causes them to move. I lost a cat recently because what was called a foxtail, a little piece of vegetable matter which has little spines on it that go back in one direction and has a sharp end at the other. It's a seed. That seed is meant to work its way into the ground by random action of rainwater, of vibrations of the earth, and just sort of movements around, because once it goes down this way easily, but doesn't go this way very easily. It's kind of a ratchet. This one got stuck in my cat's body and actually went through and migrated actually from her anus up into her chest cavity. It migrated through the body just by random movements of the digestive system and so on. That is a ratchet effect. What I want to say here is this structure I just described to you is a kind of a ratchet. It ratchets constraints. It captures constraints. But this is precisely what's needed for evolution in life. It's constraints, regularities, organization that is lost over time by virtue of the second law of thermodynamics, by virtue of the way nature tends to spontaneously fall into mess. But if you have a system that utilizes that very tendency and then captures it before it's run out, it's a ratchet. It captures constraints, and that means you can capture additional constraints. That is, you can multiply the order of it, or you can propagate the order, you can reproduce it. So what I'm arguing here is I think this is a system that's sort of the minimal system, which has a kind of self-repair, self-maintenance, and even the capacity to self-reproduce, and, as I'll say in a moment, even to evolve. This also is a process that produces what I would call true selves. The word self in self-organization doesn't carry any weight. It just simply says it's not done from the outside. But here we're talking about a self. This is because the structure itself, the dynamics of this structure and the constraints it maintains, produce itself. They are contained within it, and they are what drive the work that can be done. And in that respect, it's a self, what I would say the first very minimal concept of self. And although I'm not going to go on and talk about self in a highfalutin way, like in terms of talking about consciousness self or human self, psychological self, I want to point out that organisms are the first place where we have to talk about self. We talk about an adaptation. It's about maintaining itself. When we're talking about reproducing, we're talking about reproducing itself. Here, self does play a very significant role. It's a kind of agentive kind of role. What's happening is the synergy of the whole, and the unity of the whole is what's being maintained. And notice that in this case in which these things get broken up and reconstituted again, it's not because they're physically consistent. The physical parts can be changed in this process. The only thing that's being passed on, especially over multiple iterations, are the constraints themselves. The reason that life can produce new structure and take in new kinds of molecules and use them to do the same old thing is because it's not dependent on passing on the old stuff. It's dependent upon preserving and passing on the same constraints. But the self is a sort of higher order constraint, you might say. It's a constraint that constrains, that limits the way this can happen, that keeps these relationships between processes linked together. I want to just say, in quickly passing before I end here, I want to go on to one last interesting issue. And that is, because this is a system that is not completely closed to the world, but continually being opened up and closed, it's going to sample the world, so to speak. And if other molecules floating around in the world do a better job, they're going to tend to replace the old molecules. This has very limited evolvability, but in fact it has some kind of very, very limited and weak evolvability. Notice that there's no metabolism per se, no information molecules per se, nothing that we would think about as life as we know it. And yet it has all these qualities that we normally associate with life. Again, it's my model of what I think is the simplest version of what I want to call teleodynamics. Dynamics that has an end, that's directed towards something. Okay. So what about information? What about the information that makes up living things? There's a lot of problems with the theory of information as we use it today. We mostly talk about it in the abstract. We talk about how much information my hard disk can hold, or what the bandwidth of my home system is in sending signals back and forth to the internet. We think about it with respect to what it represents and what it accomplishes, its function, so to speak. The engineering theory that we call information theory is incredibly powerful and incredibly productive precisely because it doesn't worry about those things. But when we talk about life or mind, for that matter, what things are about is what information is for. There may be the potential in things in the world to have aboutness, that is, to have reference. But the question is, how do they get that reference? Yes, there's all kinds of variations in the world. I look at the way people are distributed in the room. I can use that variation to make guesses as how people came here, or who sits with whom, that sort of thing. That distribution is potential information. But it's not information unless it's not about anything unless I do the work to find out what it's about. The question I'm going to ask has to do with something like DNA. What is information at that level? Can we really talk about DNA being about something? Or rather, to put it another way, how is it that a molecule can come to be about what other molecules are doing? That's a very interesting question, but I think that's what we have to be able to answer with respect to DNA. So let me give you an example by just playing with this model a little bit. How would we think about how this just simple, very, very simple chemical-like process, how could we begin to think about it in terms of what I call semiosis? Not just information, but actually the representing of something. When something is represented, it's represented to something for some function. And that's one of the challenges to build a full theory. So here is this little tube autogen, I call these things. But the difference with this one, and this was suggested by some colleagues in England, imagine that the surface molecule has a shape to it that if substrates that are used for the catalysis will stick to it, they will make the surface weaker. The more that stick to the surface, the weaker it becomes, the more likely it is to break open. The problem with this kind of a system is if it gets randomly broken open, sometimes it's in a context in which there's lots of substrate for catalysis to work, and other times it's in a context in which there isn't any. And therefore, it's not going to persist. It's going to dissipate and be destroyed. What we want is a system that, in a sense, knows something about its environment. Now, I'm not talking about knowing it in any kind of mental sense. But here's a system that now is sensitive to its context in a way that's consistent with its own dynamics. That is, it's sensitive with respect to what helps and hurts it, so to speak. It has a kind of evaluation going on here. Its dynamic changes depending on its environment, and it changes in a way that is to the advantage of its dynamics, of its teleodynamics, of its persistence. What you can say is now, in effect, the number of bindings on the surface is an indication of the concentration of substrates in its environment. How is it being interpreted? It's interpreted in the change that makes in the dynamics that reconstitutes the very dynamics that can do that again. A work cycle again. As the information is now, in a sense, providing something to the dynamics that changes the way it works in such a way that it's effectively interpreting its environment with respect to its own functional requirements. Function here is, again, something that we wouldn't think about it in living terms, but it has a kind of representational feature. In effect, the assay that it's doing of the environment here is information about its environment with respect to its own dynamical predispositions. There's a big limit to this kind of evolvability, and the limit is this. Autocatalysis, where you've got a lot of reciprocally interacting molecules, don't just do what you want them to do. They all interact with each other to some extent. And the more you mix new things in, the less the opportunity for molecules that should be interacting with each other to interact with each other. Even worse, some molecules are now interacting with each other in ways that you don't want. That's what good chemistry is about. It's about making sure that only the right molecules interact with each other at the right time, in the right order. Well, the problem with the evolvability of a system as simple as I've described is that to get more complex, it gets worse at what it does. It will get slower and slower until it can't actually make itself. It has a sort of complexity upper limit. The problem is, how are you going to deal with this? The problem is, there are not enough constraints in a system like this. Complexity is a problem. So for life to work, to be evolvable, it needs what I will call molecular aboutness. Because what's happening is this aboutness, as I've just described it, so to speak, is providing constraints. New constraints on when, in this case, you can divide or not divide, when you can break up and not break up. That's what information is. And in fact, those of you familiar with Shannon's theory of information, it's all about constraint and what he calls entropy, slightly different version of the concept of entropy. But in fact, they're related to each other. So the question I want to ask now to end this is, how can a molecular structure come to embody information about the dynamic and structural integrity of the system of which it is a part? This is the problem of DNA. How DNA has come to be informative. Not about, I'm assuming it's got information, because I'm going to say that information is not an intrinsic function. It's a function of interpretation and of function itself. My fanciful story, and it is a fanciful story, though I don't think it's too far from what could be the case, builds out of a strange coincidence of life. All the energetic processes of living cells depend upon phosphates. Phosphorus is this wonderful molecule that captures a lot of oxygen on its tips, and oxygen is a very energetic molecule. And it can use this, these molecules use this to change the shape and the activity of other molecules. Energy is ferried around by phosphorus, what we call phosphates. And it's done so in a complicated molecule that has a little base component here, a sugar part here, a 5-part sugar, and the phosphates chain off the end of it. And as you chain new phosphates on the end of it, they have a whole lot more energy. They gain in energy as you chain them out there. And so you take the third one off and you go from what's called ATP to ADP, and you release a lot of energy. And that's what I'm sort of caricaturing here. You might change the structure of a protein in that process to cause its change of activity. Here's the odd coincidence. These are called nucleotides, and there are a number of different nucleotides that all play energetic roles in one way or another. Why should it be that life uses the same molecules to ferry energy around in the cell, and also to be the building blocks of the information molecules of the cell? It's a curious coincidence that the same molecules are used for these two very different functions. Why might that be true? Well, my story may not be the answer to that. Something still to contemplate. Many people have contemplated it, and we still don't have a good answer for it. But one of the things we can say about a system that produced also these energy-capturing molecules, if this is one of the byproducts of its catalysis, guess what? It would potentially speed up the catalytic reaction. And if, when the system broke open, it spewed out molecules that could capture energy and then be used to modify its proteins into a more energetic function, in effect, it would speed up the catalytic processes and potentially increase its reproductive capacity. So a system that captured energy and then used it itself, was able to use it in these ways, would have an advantage, and a kind of evolutionary advantage. So you might call this energy-assisted autogenesis in this model. There's a problem, though, and that is that in the inert state, having a lot of very energetic molecules is probably not a good thing to have. Having a lot of free phosphates with a lot of free energy available, probably pretty troublesome. So a number of people have actually suggested that maybe the origins of nucleic acids, particularly DNA, might be as a kind of way to do garbage collection, so to speak. Capture all of these phosphate molecules and not allow them to be reactive. That early on it was just energy, but we just want to capture them. How do you capture them? You get rid of water for the most part, and they begin to line up like this. Well, what's interesting in this process is that when one of these processes will close up, they're going to begin to exclude water. And so spontaneously you're likely to get what we call polymers. That is, strings of similar molecules strung together one after the other. One of the weird things about the molecules, the nucleotide molecules that make up DNA and RNA, is that these are weird polymers. They're very complicated polymers in which they have, like a non-repeating crystal, they have alternate different kinds of molecules stuck in there. So I want you to envision this as a sort of thought experiment, because I'm going to try to use this to talk about how a molecule might become about other molecules. One of the things about DNA that we know of, and here's a sort of simple depiction of it, is that it has structure. And because it has structure, other molecules bind to it. One of the interesting questions that we're exploring these days is how it is that the molecules that bind to DNA play a role in regulating it. But there are lots of molecules that bind to DNA because of its structure. What's interesting is because DNA is made up of a bunch of different kinds of nucleotides strung along here, and I've just shown a half side here. That means it's going to have different structure along its length, and we know that, in fact, that is true about DNA molecules. And that means that different molecules are likely to bind to different positions along it, especially in this relaxed state when it's beginning to close up and be inert. But that means that there's the sequence of nucleotides, which is just a random process of sort of sweeping them up, so to speak, now is a substrate for a particular sequence linearizing the relationship between a bunch of other molecules. But here's the problem. If these are catalysts, here's the problem I just mentioned to you. Catalysts in large numbers of a variety interact in all kinds of ways that are not going to be very promotive of maintaining the whole system. But if they are linearized along a molecule, those that are more likely close together are likely to be released similarly and for similar reasons. They're going to be more likely to interact than molecules that are quite distant from each other. So linearizing the binding of catalysts along a template like this randomly set up polynucleotide is a way of adding constraints to what interacts with what. What does that mean? It means that distant molecules are now maybe the ones that are not going to react with such a high probability. Those that are closer together, released more likely at the same time, more likely to react with each other. What this will produce, of course, is what I call sequence-specific selection. Now, the sequence, which is originally randomly there, just as a sort of sweeping these guys up, if it is consistent with the way catalysts ought to be constrained in interacting with each other, what's going to happen now is it's going to be a better way to reproduce that very same system. Those that are not quite in order that's appropriate to catalytic interactions are going to reproduce less effectively, less often, and less successfully. So one way to think about it is that what's happening is that there's lots of possible interactions, but by linearizing this system, this now becomes a template that can be selected, can be modified and adjusted, and different variants can be selected on the basis of how closely they bring the system towards the optimal interaction among catalysts. In that respect, the sequence now carries the constraints of the dynamical network that put this thing together. It can carry that. Now, of course, that dynamical network is also dependent upon the environment, what's out there. And so the sequence is also simultaneously carrying information constraint about that as well. I use this to say one thing, and that is, in this case, this is a part of the system. Now is, in effect, a representation, an abstract of the whole dynamics. Von Neumann talked about something as the self-description. We oftentimes think about genetics as a kind of a self-description issue. Very, very indirect. This is an indirect kind of self-description. What does it describe? Not, like a description in text would. It describes the optimal constraints. That makes it information. But what this tells us, as this part now begins to represent, in a sense, the whole of which it's a part, we have this interesting logical hierarchy that's going on here, where parts represent wholes and wholes reproduce parts, and so on. A feature that's very much different than we see anywhere else in the physical universe. What this tells us something about the concept of information, not just about life. And that's what I want to end on here. What it says is that information is ultimately based in dynamics. Information is derived from dynamics. It's informing to a teleodynamic process. It does not provide information about anything to anything else. But because a teleodynamic process has a sort of self-determination and a self-tendency to go in a particular direction, one of the things that we now have is a system that, in effect, extracts the constraints that make up that process and embodies them in a structure. By that step back, it becomes much, much more evolvable, because now just manipulating that sequence is manipulating and playing with and experimenting with, so to speak, the whole dynamics of what the self is. So the point I want to make here is that biological information is not what starts life. Biological information is implicit in this kind of unusual dynamics that life has hit upon. And it allows us, stage by stage to stage by stage, to offload and make more indirect, and therefore much more powerful, evolvable, and flexible, the representation of the process we call life. I'll stop here. I was going to say something about origins of life in the broader context, but maybe we can get to that a little bit later. Thank you very much.